And as we have started the renal physiology, so we are having the series of renal physiology, in which today we are having class 2 over the topic that is juxtaglomerular apparatus and renal circulation. So before proceeding, we should learn about some core features of Auricle. So in the core feature of Auricle, first we have the live classes. So live classes are for the elevation of the learning of the students, where knowledge means interaction, means there will be one-to-one interaction of the faculty with the students. Then also on Auricle, we provide recorded sessions in which the student will have access of the recorded classes anytime, anywhere through the recorded sessions. Then also Auricle provides notes that are basically ultimate comprehensive books that are CBME based question answers, MCQs, and also full zone assessment for the students. Then also in Auricle, we provide MCQs that are from beginner level to expert level, consist of standard MCQs. Then also the students can book a slot for personalized counseling session, in which there will be a direct interaction of the student with the counselor. Then on Auricle, we have some subscriptions plan like basic, standard, and premium. So basic plan is of 3 months, which is cost around 15,000. The services including are pre-recorded classes, live classes, quick revision examination module. For a standard plan that is of 6 months, it's cost around 30,000. The services are included that are pre-recorded classes, live class, live doubt clarification, quick revision examination module. In premium plan that is of 12 months, cost around 50,000. All the services of Auricle is included in this plan, like pre-recorded classes, live class, live doubt clarification, one-to-one personalized guidance, notes, quick revision examination module. So I would recommend all the students to take the premium plan of Auricle. Also, for 20% instant discount, you can use my code, that is Aanchal 20. Now, as we have started the series of 8 classes, so today we are having, on 11th March, we are having the class over the JG apparatus and the renal circulation. On 12th, we will have on urine formation. 14th March, we will have class on concentration of urine. Then on 16 March, the class will be on acid-base balance. Then 18 March, the class will be over the topic that is maturation. Then on 20th March, the class will be over dialysis and diuretics. So starting with today's topic, that is juxtaglomerular apparatus and renal circulation. So this is the clinical case scenario that will be discussed in the end of the class. So basically, the juxtaglomerular apparatus is a specialized organ, right, which is situated near the glomerulus. As you can see in the diagram also, this apparatus situated near the glomerulus. So actually juxta means near. So based upon its location, its name has given juxtaglomerulus apparatus, means the apparatus which is present near the glomerulus. So this juxtaglomerular apparatus is formed by 3 different cells. First, we have macula densa. Second, we have extraglomerular mesangial cells. Then third, we have juxtaglomerular cells. So as in the diagram also, we can see the juxtaglomerular cells, right? These cells are situated here. Then extraglomerular mesangial cells are present in here. And the glomerular cells are present. Glomerular mesangial cells are present here. So in the next diagram also, we can see the location of the JG cells. Here, the location of the JG cells. Then macula densa cells are present. Then we have these endothelial cells or glomerular capillaries. So this is all about the cells which are basically the part of JG apparatus, right? So one by one, we're going to discuss about them also. So first we have the macula densa. So basically, this is the end portion of thick ascending segment before it opens into the distal convoluted tubule. It is situated between afferent and efferent arteriole of the same nephron. So as in the diagram also, we can see, we have seen it is very close to the afferent arteriole, and macula densa is formed by tightly compact cuboidal epithelial cells, right? Now, this macula densa is also responsible or plays a very important role in tubuloglomerular feedback. That also we're going to learn. And also it secretes a very important hormone, that is thromboxane A2. Now this thromboxane A2 causes renal vasoconstriction, and this hormone is responsible for decreasing renal blood flow also. Right? So this is all about the macula densa. Then next we have the extraglomerular mesangial cells. So these cells are situated in the triangular region bounded by afferent arteriole, and macula densa. So as in the diagram also, these types of cells are situated here. Okay? So means basically these cells are situated in this triangle bounded by afferent arteriole, bounded by efferent arteriole. Okay. Another thing, that these cells also secrete a very important hormone, that is prostaglandins, which also responsible for decreasing blood pressure. Right? Now, the third we have, and a very important one, that is JG cells. Okay. So these cells are specialized smooth muscles cells situated in the wall of afferent arteriole just before it enters the woman's capsule. As in the diagram, we have seen the location of JG cells here. Right? This is the location of JG cells. Now, these smooth muscle cells are present in tunica media, tunica adventitia of the wall of the afferent arteriole. So this is the location of the JG cells. Now, the JG cells are also known as granule cells because of the presence of secretory granules in their cytoplasm. So some type of granules are present in the cytoplasm of JG cells. So that's why the another name for JG cell is granule cells. Right? Now we will learn about the very important function, and firstly we will learn about the secretion of hormone, right? So the primary function of the juxtaglomerular apparatus is the secretion of hormone. So basically two hormones are secreted by this apparatus, that is first is renin and the second is prostaglandin, right? So one by one we're going to discuss. So the juxtaglomerular cells secretes renin, and renin is a peptide with 340 amino acids, and along with angiotensins, renin form the angiotensin systems or RAS system. So which is a hormone system that is very important in the maintenance of blood pressure. So today we're going to learn about the RAS system, that how the renin is a very important stimulator for RAS system that we're going to learn. Now, first of all, we should learn that what are the stimulants for renin secretion. Means what are those conditions in which there will be secretion of renin will occur from the kidney. So whenever there will be fall in arterial blood pressure, when there will be reduction in the ECF, or any increase in the sympathetic activity, or decreased load of sodium and chloride in macula densa. So in these conditions, there will be release of renin, right? So this is the diagram, and this is the this flow chart is basically showing the whole RAS system. Okay, so this is renin, angiotensin system, right? So, as we have discussed that these are the stimuli that is responsible for the secretion of renin from the kidney, in which we have learned whenever there will be low blood pressure or low ECF volume or increase in any sympathetic stimulation or low sodium, low plasma sodium level, right? So what will happen whenever renin is released into the blood, right? So from JG apparatus, the renin will secrete it into the blood. It acts on a specific plasma protein, that is angiotensinogen, or this is also known as renin substrate. Okay, so whenever renin is released into the blood, it will act on a very specific substance, that is angiotensinogen, or this is also known as renin substrate, or this is a type of alpha 2 globulin, right? So by the activity of this renin, this angiotensinogen get converted into a DACA peptide, right? And this DACA peptide is angiotensin 1. Okay, so this angiotensinogen with the stimulation of renin is converted into angiotensin 1. This is a DACA peptide. Right? Now, what will happen? This angiotensin 1 is converted into angiotensin 2, which is a octapeptide by the activity of a very important enzyme that is released by lungs, and that is ACE. And here ACE stands for angiotensin converting enzyme, which is released by lungs. So, this angiotensin is converted into angiotensin 2 in the presence of this important enzyme, which is released by the lungs, right? So this angiotensin 2 is a octapeptide. Okay. So basically, most of the conversion of angiotensin 1 to angiotensin 2 takes place inside lungs with the help of this enzyme, ACE. Now, AC. Now, angiotensin 2 has a very short lifespan, right? Of about 1 to 2 minutes. So what will happen after 1 to 2 minutes of the conversion of angiotensin 1 to angiotensin 2? This start rapidly degradation, means the degradation of angiotensin 2 will start, and finally it will convert into angiotensin 3 with the help of a very important enzyme, that is angiotensinases. Okay. Now, basically, this is present in some RBCs in vascular beds of many tissues, right? Finally, this angiotensin 3 get converted into angiotensin 4, which is a hexapeptide. Okay. Now, one by one, we're going to learn about the role of these hormone, that is angiotensin 1, angiotensin 2, 3 and 4. One by one, we are going to discuss that what is the main function of these hormones and what is the significance. Okay. So, first we have angiotensin 1. So basically, this is the physiologically inactive hormone, and it only serves as a precursor for angiotensin 2. Okay, so always it remain physiologically inactive. Right? Now, next we have angiotensin 2. So the work of this angiotensin 2 is very important, and it is the very means it is the most active form, and its actions varies on blood vessels, second on adrenal cortex, Then on kidney, on brain. So one by one, we're going to learn the action of angiotensin 2. First, we will discuss the action of angiotensin 2 on blood vessels. So basically, this angiotensin 2 increases the arterial blood pressure, right? By directly acting on the blood vessels and causing vasoconstriction. So, this angiotensin 2 is responsible for increasing the arterial blood pressure on the blood vessels by causing the vasoconstriction, and it is also a very important constrictor for arterioles, right? Now, it also increases blood pressure indirectly by increasing the release of another hormone that is known as noradrenal from post-ganglionic sympathetic fibers. So by increasing the secretion of this hormone, again, the blood pressure will increase because this is also a vasoconstrictor, right? So this is all about the action of angiotensin 2 on blood vessels. Now next we have on adrenal cortex. So basically the angiotensin 2 will act on the zona glomerulosa of adrenal cortex to secrete a very important hormone that is known as aldosterone. Now, this aldosterone will act on renal tubules and it increases the retention of sodium. So, whenever there will be retention of sodium, so directly the blood pressure will increase, right? Now we will next learn the action of angiotensin 2 on kidney. So basically angiotensin 2 regulates GFR, right? So we will learn that how the GFR will be regulated by this angiotensin 2. So basically this angiotensin 2 is responsible for the constriction of efferent arteriole, right? This is responsible for the constriction of efferent arteriole, which will result in decrease of filtration, right? Then also this angiotensin 2 will causes the contraction of glomerular mesangial cells. Okay, so angiotensin 2 is responsible for contraction of mesangial cells, leading to decrease in surface area of glomerular capillaries and results in decrease of filtrate. So in this way, there will be decrease in filtration also. Right? Now we have learned the action of angiotensin 2 on blood vessels, adrenal cortex and kidney, and now we're going to discuss the action of angiotensin 2 on brain. So basically this angiotensin 2 is responsible for inhibition of baroreceptor reflex, right? So whenever there will be inhibition of baroreceptor reflex, so basically thereby directly increasing the blood pressure, because this baroreceptor reflex is responsible for decreasing the blood pressure. But if there will be inhibition of this baroreceptor reflex, so ultimately it will lead to increase in BP. Okay. Another thing also, that this angiotensin 2 also causes increase in ADH, right? So if there will be increase in ADH, so means there will be increase in water reabsorption, right? Then also, angiotensin 2 is also responsible for increasing the secretion of corticotropin releasing hormone from hypothalamus, CRH, right? So angiotensin 2 is also responsible for increasing corticotrophic releasing hormone, which ultimately increasing the secretion of ACTH from pituitary, means increasing the secretion of adenocorticotropic hormone, right? So in this way, the angiotensin 2 is very important factor for increasing the blood pressure or decreasing the filtration rate. Now, next we are going to learn about the action of angiotensin 3. Okay, so basically angiotensin 3 is responsible for increasing the blood pressure and also stimulates aldosterone secretion from adrenal cortex. So this angiotensin 3 is responsible for increasing BP and also stimulates the secretion of aldosterone from adrenal cortex. Okay, now last we have angiotensin 4. This angiotensin 4 also has adrenocortical stimulating and vasopressor activities. So basically, in this way, it is also responsible for increasing the blood pressure, right? So we have discussed all about the angiotensin 1, 2, 3, and 4. Now back to the chart. So as we have discussed, whenever there will be low blood pressure or low ECF, or there will be sympathetic stimulation, low plasma sodium. So there will be secretion of renin, means some things should be happen so that these all things should get back to normal. So this mechanism will be there in which angiotensin 2 is responsible for the regulation of GFR, increasing the blood pressure back to normal, increasing water intake, increasing ADH secretion, increasing CRH and ACTH secretions, so that all the things which get very low, they will get increased and become back to normal, right? Now, the last, we have angiotensin 3, as we have seen that this hormone is responsible for increasing the secretion of aldosterone and also causes vasoconstriction. So this is all about the RAS system. I hope this is very helpful to the student. One more flowchart we are having for RAS system, in which they have shown that whenever there will be decreased arterial pressure, so there will be increased stimulation of JG cells, which causes the secretion of renin into the blood. Then renin cleaves the plasma protein, that is angiotensinogen, which inactive decapeptide angiotensin 1 is formed with the help of this. Then angiotensin converting enzyme on lungs capillary clips this to angiotensin 2, as we have seen. Now this angiotensin 2 raises the systemic blood pressure. We have seen by several mechanisms it increases. Then angiotensin 2 stimulates the adrenals to secrete aldosterone, and aldosterone promotes sodium and water reabsorption in the DCT, and in this way finally the blood pressure will increase and become back to normal, right? Now next we're going to learn about the other secretions. So Once again, we're going to revise about the RAS system. So whenever renin is released into the blood, it acts on a specific plasma protein known as angiotensin or renin substrate, which is the alpha-2 globulin. By the activity of renin, the angiotensinogen is converted to a DACA peptide known as angiotensin 1, and angiotensin 1 is converted to angiotensin 2, which is the octapeptide, as we have seen by the activity of ACE, which is secreted from the lungs, and most of the conversion occurs in lungs. Now, this angiotensin 2 has a very short lifespan of about 1 to 2 minutes. So, again, it will converted or degraded into angiotensin 3, which is present in RBCs and vascular beds in many tissues. Now, this angiotensin 3 is converted to angiotensin 4, which is a hexapeptide, right? Now, next we're going to learn, okay, one more thing that as we have discussed, this aldosterone is secreted by the stimulating activity of angiotensin 2, which increases potassium concentration in plasma and also causes reabsorption of sodium, secretion of potassium, occurs at DCT and collecting ducts, right? Now, another thing we have the prostaglandin, the role of prostaglandin, which is also secreted by this JG apparatus, right? So, this type of cells, basically the extraglomerular mesangial cells of juxtaglomerular apparatus secretes this prostaglandin, okay? And this is responsible for decreasing BP by vasodilation, okay? And this prostaglandin is secreted by interstitial cells of medulla, which is known as type 1 medullary interstitial cells, right? Okay, then secretion of other substances also there, like extraglomerular mesangial cells of juxtaglomerular apparatus secrete a cytokine like interleukin-2 and some tumor necrosis factors. And also macula densa also secretes a very important hormone, that is thromboxane A2, which is responsible or also causes renal vasoconstriction, means decreases renal blood flow, right? Okay, now we're going to learn about the renal circulation. So as we know, the blood vessels of kidney are highly specialized to facilitate the functions of nephron in the formation of urine, right? So in the adults, during resting condition, both the kidney receive 1300 ml of ml blood per minute, which is about 26% of cardiac output, right? So maximum blood supply to kidney has got a very significant functional significance, because renal arteries are supply blood to kidney, and renal arteries is the direct branch of the aorta, right? This is that is also we're going to learn. So let's discuss about the circulation. Okay, so renal regulation of renal blood flow. So basically, as we have discussed, the renal artery is the direct branch of the abdominal aorta. So this renal artery arises directly from this abdominal aorta and enters the kidney through a structure known as hilus. While passing through the renal sinus, the renal artery divides into many segmental arteries, right? So this renal artery gives rise to many segmental artery. Now, this segmental artery will subdivide into many interlobar artery, right? So, whatever the amount of blood that is received by segmental arteries will drain into interlobar artery, right? Now, these interlobar arteries passes in between the medullary pyramids that is present in kidney. So, this interlobar artery will passes in between the medullary pyramids. So, at the base of the pyramids, it turns and run parallel to the base of pyramid, and there it forming the arcuate artery, right? So from interlobar arteries, there will be formation of arcuate artery. Now next, from the arcuate artery, each arcuate artery give rise to interlobular artery. So always remember interlobular and interlobar, both are different. Okay. So from arcuate, there will be formation of interlobular artery, right? Now, these interlobular artery run through the renal cortex and penetrates to perpendicular to arcuate artery, and from each lobular artery numerous afferent arterioles are formed. And as we have seen in the structure of nephron also, this afferent arteriole is responsible for the blood supply of each nephron, right? Now these afferent arterioles enters the Bowman's capsule and forms the glomerular capillaries, right? After entering the Bowman's capsule, the afferent arterioles divide into 4 or 5 large capillaries, right? And finally, each large capillaries divide into small glomerular capillaries and forming the loop, as we have seen in the structure also, forming the loop. And the capillaries loops unite to form these afferent arteriole, which leaves the Bowman's capsule. So there will be supply of blood from afferent arteriole to glomerular capillaries, as we have seen in the diagram of nephron. Or if you have not seen, you can watch my last lecture. And from glomerular capillary, there will be formation of the loop-like structure, which unite to form the afferent arteriole, right? Now, from efferent arteriole, what will happen? From this efferent arteriole, from the second capillary network that is known as peritubular capillaries, right? Which surrounds the tubular portion of the nephron, as we have seen in the last lecture, right? And the renal circulation forms a portal system by the presence of two sets of capillaries. The first we have peritubular, and second we have glomerular capillaries, right? Now, we have peritubular capillaries and vasa recta. So, basically, these peritubular capillaries are found around the tubular portion of cortical nephrons only, and the tubular portion of juxtamedullary portion are supplied by some specialized capillaries, which are known as vasa recta. As we have discussed in the last class also, that these peritubular capillaries are found in cortical nephron, whereas vasa recta are found in juxtamedullary, right? Now, the vasa recta arise directly from the efferent arteriole of the juxtamedullary nephron and runs parallel to the renal tubule into the medulla and ascends up towards the cortex. Okay, then what will happen? There will be drainage of blood into the peritubular venule, and again from the venule the blood will drain into interlobular vein, from interlobular vein then to arcuate vein, and from arcuate vein to interlobar vein, then from interlobar to segmental vein, then forming the renal vein, then finally back to inferior vena cava and back to heart, right? So this is all about the regulation of renal blood flow that is occurs inside kidney, right? Now next we have the auto-regulation of kidney. So basically, this auto-regulation is important to maintain the glomerular filtration rate. This renal auto-regulation is important to maintain GFR. Okay, so blood flow to kidneys remain normal even when the arterial blood pressure rises. Okay, so when the mean arterial blood pressure varies between 60 to 180 mmHg, so in this way the GFR remain maintained, because the kidney has the very important property of auto-regulation of BP. Okay, so here we're going to learn about two important mechanisms. The first we have myogenic, and the second we have tubuloglomerular feedback. One by one we're going to learn. So first we have myogenic mechanism. So what happened whenever the blood flow to kidney increases, there will be stretching in the elastic wall of the afferent arteriole. Okay. So whenever there will be rise in blood flow, okay, so there will be stretching in blood vessels, yeah, vessel wall of afferent arteriole, right? So if there will be stretching of the blood vessels walls, so thereby increasing the flow of calcium ion from ECF to the cell. So means this stretching will increasing the movement of calcium ions from ECF to vascular smooth muscle cells, right? So now this calcium will combines with calmodulin, and there will be activation of myosin cross bridge. And with the activation of this myosin cross bridge, there will be contraction of vascular smooth muscles, and there will be increase in renal blood flow and GFR, means both will back to normal. Okay, so in this way the myogenic mechanism works. Again, I'm going to repeat. So whenever the blood flow to kidney increases, it stretches the elastic wall of afferent arterioles, and stretching the wall of efferent arterioles, increasing the flow of calcium ion from ECF into the cells, and with the help of or with the activation of myosin cross bridge, the influx of calcium ions leads to the contraction of smooth muscles basically, and which causes the constriction of the afferent arteriole, and finally there will be decrease in the blood flow and GFR, and both will become normal, right? So this is all about the myogenic mechanism. Now next we have the tubuloglomerular feedback. So first we're going to discuss. So basically in tubuloglomerular feedback, there is a work of very spatial cells, that is macula densa. Basically this is responsible for controlling the renal blood flow and GFR, and how that we're going to learn. Okay. So what happened whenever there is increase in renal blood flow or increase in GFR? So ultimately, there will be increase in sodium ion concentration in lumen, right? So this is sensed by macula densa. And in this response, it will secrete a very important substance, that is adenosine, which act on smooth muscles of afferent arteriole and causes vasoconstriction. And there will be decrease in GFR and decrease in renal blood flow, means they will get back to normal. So basically this adenosine is responsible for this action, which is secreted by macula densa whenever there will be increase in the concentration of sodium ion due to increased renal blood flow and increase GFR. Okay. And again, there will be vice versa to this. Whenever there will be decrease in renal blood flow or decrease in GFR, so ultimately there will be decrease in sodium ions also, in lumen. So this is also sensed by this macula densa, and ultimately there will be role of adenosine, causes vasodilation, and there will be increase in renal blood flow and increase in GFR. Okay. So this is all about the tubuloglomerular feedback. Okay, now talking about some special features of renal circulation. So renal circulation has some special features to cope up with the function of the kidney, and we're going to discuss. Like the renal artery arises directly from the aorta. This is the direct branch of the aorta. So the high pressure in the aorta facilitates the high blood flow to the kidney. This features already we have discussed. Now the next feature, both the kidney receive 1300 ml of blood per minute, which is about 26% of cardiac output. So, kidneys are the second organ to receive maximum blood flow. The first is the liver, which receives 1500 ml per minute, right? So about 30% of cardiac output. So basically the kidney receives maximum amount of blood. Now the third, when whole amount of blood which flows to kidney has to pass through the glomerular capillary before entering the venous system. So because of this, the blood is completely filtered at the renal glomerula. Okay. And another feature we have that already we have discussed, the feature of autoregulation. Right. So these are the some special features of renal circulation that occurs inside the kidney. Right. Now we have some MCQs. So I'm giving 30 seconds to all the students to solve this MCQ. The first is the very easy one. Which of the following cells releases renin? And the options are extraglomerular mesangial cells, macula densa, alpha intercalated cells, or JG cells. You have 30 seconds to give answer. Yes, the answer is D. Now, macula densa cells sense concentration of which of the following? Just now we have discussed this question: ADH, ANP, calcium, or sodium ion. Macula densa sense what? Again, you have 30 seconds to answer this. Yes, the answer is sodium ions. Now the last, an increase in the concentration of plasma sodium causes increase in what? Release of renin, secretion of aldosterone, secretion of ADH, release of ANP. What is the answer? Yes, the answer is 1, release of renin. Okay, back to the clinical case. So a 28-year-old female present to the clinic with complaint of persistent high blood pressure and occasional episodes of dizziness. Upon further investigation, the healthcare provider suspect a dysfunction in the JG apparatus. So which of the following cells in the JG apparatus release renin in response to decreased blood pressure or low sodium level? So your options are macula densa, mesangial cells, podocytes, or juxtaglomerular cells. Yes, the answer is T, juxtaglomerular cells. So this is all about the juxtaglomerular apparatus and renal circulation. I hope all the students find this class useful for them. Thank you so much.